everyone, and welcome to another edition of What's Up with FOXG1 Research. Today we have a new exciting edition for you. We are joined, as always, with Nasha Fitter, my co-founder, and our CEO, and Ryan Phillips, our CFO. And today we are joined by a very special guest, Hourinaz Behesti. She is our new Director of Science. Hourinaz is a developmental neurobiologist from Rockefeller University. She has a long-standing interest in brain development and neurodevelopmental disorders, and she's been studying the function of autism risk genes, which FOXG1 is, and has developed models to study the development of human brain cells in a dish. So we're so glad to have you here, Hourinaz. She's been on our SAB since the beginning, and now we could officially call you our Director of Science, which is so exciting. So our first question we want to lead with is, what brought you to us? What brought you to FOXG1?
Well, thanks, Nicole. Thank you for having me. FOXG1 is a really interesting gene as a developmental neurobiologist. It's one of the earliest genes that is expressed, and it sort of like puts the blueprint for how the brain is going to get developed, and it sort of tells which part of the region, which region of the brain is going to become the forebrain, which gives rise to the cortex and sort of higher functioning areas of the brain. So as a developmental biologist, I've always known about FOXG1, and I also studied it briefly, actually, when I was doing my PhD in eye development, because it's also expressed in the eye, in the retina. And yeah, a few years ago, when you guys had just sort of gotten started, Nasha and I were put in touch through a mutual friend. And through conversations, we just realized that my expertise is really suited for what you're trying to do and for the mission of FOXG1, and for trying to sort of help you with the research strategy. So that's how I became involved with the SAB back in 2017.
And at the time, there just wasn't that much known about FOXG1. So what were your thoughts on that?
Well, there have been a few sort of basic science studies in mice. So it was a little bit known about FOXG1 biology from that point of view. But as you all know, just FOXG1 syndrome wasn't really diagnosed. What is it now? Has it just been like 10 years now? A little over 10 years. And so there's really not much known about FOXG1 in the context of disease and FOXG1 syndrome. So yeah, I think what the foundation is doing is really amazing in terms of trying to cover both the basic science of FOXG1 and try and understand FOXG1 biology a little bit more in depth, but of course also try and understand the disease biology. So
that actually leads me to a question I really would love for us on this call to break down. And that is, what is our foundation's research strategy? Because that's something we really want to approach with a strategy so that we can get to our end goal, and what's the best way to do it. So since you've come on board and you've been on board, how would you define the strategy that we're taking and why we're taking this strategy to really leave no stone unturned?
So of course, the big aim of the foundation is to try and find a cure. But to be able to find the cure, you really need to understand the biology in a little bit more detail and in a bit more depth. And I think that's one of the strengths of the FOXG1 Foundation. And I'm so amazed that you guys as parents came together and also realized this early on, that you need to understand the gene and the basic biology of it a little bit better. And so there are lots of projects that are aimed at basically just understanding FOXG1 biology, but also in the context of the patient mutations. So one of the things that you guys did very early on, which was really impressive, was to fund the making of a number of mouse models that sort of cover a wide range of mutations that are seen in the children. And so that work is progressing really well. I think it's most of the mouse models are ready now, and there's quite a lot of analysis that's been done by the labs who are working on them. And then at the same time, so that's really exciting because that allows you to actually look at the patient mutations in an organism, which is really important because you'll be able to also understand the effects of sort of environmental effects and also just how at the organismal level what the mutations behave like. But of course, mice are not humans. And so eventually you would want to study them also in a human context. And for that purpose, the foundation's taken advantage of this new technology called iPSC technology, which allows you to turn patient cells or any kind of cell from a patient. I think in the case of FOXG1, you have mostly been collecting fibroblasts from patients. And so you can actually turn them using this amazing technology, which was awarded the Nobel Prize actually back in 2006. So you can turn them into any other type of cells. So in the case of FOXG1, it makes sense to make them into nerve cells and into sort of other support cells of the brain. And so there are a bunch of projects that are on the way to specifically study also the patient mutations within the context of this system. So actually look at human nerve cells and try and understand the biology of it.
Okay, so FOXG1 is different because there's so many different variants of FOXG1 syndrome. It's not homogeneous. There's, Josie has a nonsense mutation, Amara has a missense mutation. So, can you talk a little bit about our strategy and how we are approaching this so that we can come up with treatments for all children with FOXG1 syndrome?
Right. So one of the things that as you said, is really apparent in FOXG1 syndrome is that different patients have different manifestations of the disease. And so it was apparent, I think, from very early on that different mutations might actually be causing these different, what we call phenotypes. And so the strategy of spending the time to develop lots of different models to kind of cover the spectrum of mutations that are known in FOXG1, in the FOXG1 community, has actually been really useful, I think, and is starting to bear fruit now because the models, the time and the resources have been spent and the models have been created, and the sort of initial analysis is suggesting that yes, there is variability in phenotype across these different mutations. And it's a really important asset to have to then be able to also compare it directly to the patient phenotypes through the natural history study. And I think that together that's going to be a really powerful platform for bringing the science forward. We're also looking at screens. So so this is a very exciting area, I think. So this is basically sort of like a shot in the dark. So we don't really depend on completely understanding the biology or the disease mechanism. It's more about utilizing the assets that the foundation has already created to then screen a bunch of drugs. So for example, a bunch of FDA approved drugs that are already out there. And you can do that in what's called a high throughput fashion. So you can screen something like 3,000 drugs in a system. And so what I have been working on with the foundation is to try and get 3 different kinds of screens off the ground. Mm-hmm. So one is in a zebrafish model, so it's a fish. But as biologists, a lot of these sort of mechanisms are highly conserved, and so what we do is that we go to simpler systems. We try to minimize the complexity and go as simple as possible. And so one way of doing that is to use a zebrafish model because it's easy to grow, and they also sort of share certain features that that we think are also happening in the patients. And so through this high throughput screen, they can screen something like 3,000 FDA approved drugs in a matter of months and identify anything that might actually make the mutation or the mutant sort of behavior turn back into a normal behavior.
Another see that on a zebra, and as we would say zebrafish. What would a zebrafish show you? So you were talking about the different models that are developed of FOXG1. There's mouse models, iPSC lines, which we'll get more into that as well, and zebrafish models. So if you got all these drugs, we're going to screen. So you're saying that the zebrafish is sort of just like a very simple test to see on a simple level of the changes that happen in the fish?
Exactly, but its brain sort of is the sort of basic architecture of its brain is very similar to the basic architecture of a human brain. Mm-hmm. And so there are very similar cell types that exist in the human brain that also exist in the zebrafish brain that we know are impacted by FOXG1 mutation. And so what we can do is that the lab that is actually carrying this screen out, what they have done is that they have made sort of markers that light up specific nerve cells in the brain of a zebrafish, and then that allows them to by microscopy just see them. And
right. We've seen incredible images from Corinne Hart's lab of these Christmas tree zebrafish brains. This is an example of what this looks like when you do this.
Exactly. So this is it's a very powerful type of screen because it's what we call a phenotypic screen, and so it sort of looks at the output of like everything that might be wrong in the brain of a FOXG1 patient. It sort of like picks one of those things and then looks for that thing and see whether we can correct it by adding these drugs.
Okay, so phenotypic screens. That's one of the screening projects. So going through, thank you for going through sort of the strategy that we have, and would you consider it like a holistic approach to our research strategy? All these different modalities, right?
Yes, exactly. I think the bigger picture, of course, the bigger aim is to find a cure, and to get there, you need to have lots of different branches going on at the same time. And so one branch is to understand the disease biology better in the mouse models and the iPSC lines, and then the other strategy is to find drugs sort of in a shortcut way, which is what these screens are. There are also, so there's one more screen that I want to talk about as well, which is another phenotypic screen so instead of doing it on fish, this is actually on human cells. So that's the strength of it. But the problem with human cells, well, it's a problem in all sorts of experiments, but this is a really big problem in human cells, is that there's really a lot of source of variability when you're trying to model something in a day. So even though we know we have sort of protocols for making nerve cells, and every time we use them, we get nerve cells, but there's always like a level of variability, and that sort of confounds the interpretation of the data and the understanding of the data at the end. And so what this company that has actually spun out of Rockefeller University called Rumi Scientific have done is that they've overcome this limitation by coming up with a really clever screening platform where they sort of minimize the variability. So I can show you a little bit what it looks like here by sharing my screen. Can you see?
Mm-hmm. And this is a project we have underway. So this is something you're excited about as part of our...
Yes, exactly. This is a new project that is underway and that we do in collaboration with Rumi Scientific. And so this is basically images of, and this is again another phenotypic screen, but it's on human cells. And so these are images of what the patient nerve cells will look like when they're grown on their platform. So let's see, the first one is sort of just a normal subject, so no mutations. And then you can see how sort of eye-catching it is when they try to model different conditions that are actually very related to FOXG1 syndrome, so Fragile X and Rett syndrome. So without going into detail about what we're looking at here, you can just, I'm sure, visually appreciate how you can very easily pick out what we call a phenotype, which is basically that it looks different from the normal state. And this is, so that was looking from the top down at these neural structures, which they actually call neuraloids. And this is looking from the side. And so again, you have no mutation, you have Fragile X, and then Rett syndrome. And you can again see how they look very different between the three different conditions. And then what they have developed is a sort of like a AI-powered imaging screen that they can then use to just image them, and then the AI will identify, okay, this is abnormal to this category, to this strength, and then they can use again a library of compounds to screen. So, for example, the 3,000 compound rich FDA approved library. And so here's another example that they have done with Huntington's disease. You can see how messed up the Huntington's disease one looks like. And then here is the Huntington disease rescued with a drug candidate that they identified. So
wow.
You can appreciate how it's sort of like, it's very easy to detect how something can go back to normal. And this is without really understanding too deeply exactly mechanistically what is happening.
And this is what we call a hit. So when we use the language, if we get a hit, this is a hit.
Exactly, exactly.
Right. Hourinaz, that was really, the last comment you just made was really insightful to me when we just, we had this discussion a couple of weeks back, which is, we're funding leading neurobiologists understanding FOXG1 very acutely to intervene with, tactical ways of how exactly we would correct FOXG1 once we understand it. But there's also this approach where let's just look at abnormalities, phenotypic abnormalities, and drop a ton of drugs on it and see if they make it look normal or correct that abnormality that's visually apparent, as we see here. Mm-hmm. And kind of back into what is the mechanism causing the problem. So you kind of fast track to let's solve the problem before maybe we fully understand what caused the problem, which is really an interesting approach when we have both, going down both routes. Could you talk a little bit more about that?
Yeah, I think it's a really important approach, and science has progressed enough to be able to actually do this in human neurons now, which is what basically this screen is going to allow us to do. And hopefully it will yield, I'm confident that it will yield some hits, basically, as Nicole was saying. And as you said, this is a way of sort of bypassing needing to know too much about the system, but sort of visually just be able to revert it back into a normal state. So I think this is one strategy, but I think the other strategy of actually trying to understand the disease and understand the biology is also equally important, that needs to be taken at the same time. Because this is sort of like a candidate approach strategy, whereas the other strategy would be much more informed. We will be able to figure out exactly what to go after. And this is sort of like a shot in the dark kind of way. So by doing these two different threads, I feel like all the bases are covered with what is available in terms of technology at the moment that can be utilized.
Okay, so these are phenotypic screens. So what's the other type of screen that's important to see the whole picture?
So there's one more screen that the foundation is pursuing, which is basically a transcriptional screen. And that will actually So in a nutshell, what the information that gives is information about gene expression. So how the different genes in a cell are expressed. So we can look at all the different genes that are expressed in a nerve cell at the same time. And then we can ask the question of, if we modify FOXG1 and make it into like a patient mutation, how does it change? How does the overall architecture or landscape of gene expression inside of the cell change? And then if you put back these drugs, again, how does it change? Are we able to what we call a rescue of a phenotype? And so that screen is sort of gives you a very different kind of information. And in many ways, it actually gives you more information. So not only does it identify the target hit, but it also gives you information about the expression of all these other genes inside of the cell. But it's still something that is a little bit further removed from actually rescuing what we call a phenotype in terms of, you know, what the brain would look like, how the cells sort of connect together, and is it going to be able to make the cells sort of connect in the correct way again. So a transcriptional screen is not going to be able to answer that question. So it would be that you would then identify the compound, and then you would test it further in something like this to again look at a phenotype and look at things at a cellular level.
So it's great to have both of these projects happening at the same time. It just gives us more information of, you know, what's happening and how we can then take that and look at the phenotypic screen with the same information. Yeah.
Exactly. I think it gives different kinds of information.
What's that?
It gives different kinds of information.
Right, right.
Yeah, Nicole, one way families can think about it is that we're developing our own personalized screening platform for kids with FOXG1, because right now when our children have seizures or a movement disorder, we go to our doctors and they give us non-specialized drugs, right? They give us drugs that they hope will work for FOXG1 because they work for seizures, but they've never been tested on FOXG1 mouse models or stem cell lines or any of these things. So our kids end up becoming the guinea pigs. What screens like this allow us to do is actually gather some information, and then we can make better choices on what is the right medication to give our own children. So that's one way to think about this as well.
And seeing what, you know, medication our children are on, we can actually see it in this way. So it's, you know, instead of having been the guinea pigs all this time, we could see the actual difference in cells. That's great. Thank you so much for that explanation. And we're, you know, kicking off this projects right now. What are the goals and timelines for these projects? When can we expect to have some information to share with our community?
You know, science is a slow progress. I'm not going to lie. So things take time, but they are underway. And I think that the analysis of the different mouse lines is sort of progressed really well. And there's a lot of information that is coming out of that that is really useful and can sort of inform the next steps. These screens are just starting. The phenotypic and the transcriptional screens are just starting, and hopefully within the next year or so, we should be able to have information to share, and it should be possible to have actually screened some libraries using these platforms already within the next year, I think.
That's exciting.
There's a lot of work in progress.
Right. Which is
Can you talk a little bit about the progression of what we would do with the hits and how we've kind of strategically positioned ourselves to develop the assets in place already to move forward with hits if we find them?
So, I mean, one way to test the hits afterwards would be to test them in the mouse models that have already been created. So, and the good thing about having spent time on analyzing them and understanding the biology in these mouse models is that once you have a compound, then you know exactly what to look for and exactly, you know, what needs to be corrected. So a lot of sort of like the groundwork of what we call characterization is already being done. And so if a compound was to be identified, I think the next step would accelerate quite fast, actually. And the other assets that can also be utilized for validating these drugs is the patient cell lines from the iPSC cells, basically. So these two together, and then I think there are a lot of partnerships, in place that I think Nasha can speak to more clearly about how to take this further into the clinic.
Yeah, because you've met with all our partnering labs, and now we see the process. This is everything we have in place, and now as we start to get hits, what's the next steps? How do we get this to the clinic? So what do you see as the big approach? Nasha, did you want to speak to that?
Yeah, I mean, there's some things that we may be able to actually get a compassionate use case from our children's own neurologists, depending on what that drug has already been approved for and utilize right away. And then there's other ones that we're going to have to do potentially further formulation or submit an IND an IND submission to the FDA. So I think it just depends on like what we are going to find. And the goal with this process is to try to get some sort of list of what are the best drugs for certain phenotypes in some sort of order. So we even know what to continue pursuing. But that's the goal, is and the reason we're trying to have, the foundation own as much of this process as possible is because we're not interested in developing a drug and making money. We're interested in how do we get a drug quickly to our own children. So the steps that we take, the people that we're going to be working with, the investments we're going to be taking will be completely focused on that end goal. But we'll know more as we get the results.
And that's one of the upsides of using sort of FDA approved libraries to do these screenings, because that's sort of, these drugs have already been approved, and so a lot of like the toxicology and all of that is out of the way.
Yep.
Which is great. And so it's so exciting to know that we have this path ahead of us. But what about non-FDA approved drugs? What's our strategy as far as gene therapy when it comes to correcting a gene? That's part of our approach. And so where are we? Tell our community about where we are as much as you can, as far as can we fix a gene?
Yes. So the approach to fix a gene is essentially called gene therapy. That's what gene therapy is. So you go in and either try to fix the gene that is already in the patient, or you give an extra copy of the gene to the patient. And there are several projects that are focusing on that. And there are sort of, what we call vectors. So these are viruses that need to deliver the gene therapy to the brain. And so the first step is to test them and see if they work. And if they then work in mouse models, then you try to do the same thing in the patient cells and see whether you can restore the expression of the gene. Mm-hmm. So that's one strategy. Another strategy is, so genes basically have products, right? So genes first produce something that is called mRNA. And then this mRNA is sort of like the blueprint for what gives rise to proteins. And proteins are the building blocks of cells. So it's really like a three-step way. And so gene therapy usually deals with the DNA level. And then there are sort of these newer modalities that have been developed over recent years that are referred to as ASOs or ASO therapies. And these are more targeted towards the mRNA level. So they're sort of in between the DNA and the protein level. And the foundation also has several projects pursuing ASOs as potential therapies for basically targeting the FOXG1 mRNA expression. And then we're also thinking about other strategies to actually target the protein. And this is an sort of exciting field in science in general. There's been a huge explosion in the field of it's called targeted protein degradation, which could be a really good option for FOXG1 as well. So, again, it's sort of like shot in the dark. You don't know what's going to work. You have to kind of keep at several different, strategies and see which is going to pan out the best.
So tell me, I mean, you've been in science for quite a while now, and you must have seen so many changes over the past years, right? Over the past 5 years, what would you say have been some of the biggest advancements that you've seen that have gotten you excited? And where do you see science going in the next 5 years? So maybe start with, what's been so exciting for you in your career as of recently in science? And then talk a little bit about what excites you to work with going forward or see happen moving forward.
Yeah, I mean, there's a lot of new technology that has sort of sprung up, and a lot of really important findings come because technology moves forward so that you can actually discover things that you weren't really able to get at before. And so some of the really exciting technologies that I think we have all experienced as sort of, globally as humanity is the advance in mRNA technology, which is the basis for the Pfizer and the Moderna vaccines. And so being able to deliver information in the form of mRNA to patients is now a real possibility. So that's really exciting. And this is something that can be I think tested also within the space of neurodevelopmental disorders and FOXG1 syndrome. The other exciting, it's all sort of like technology related. So, so this whole idea of the targeted protein degradation is actually something that I came across back in 2014 when I was working on this autism risk gene called astatine-2, and it's a humongous gene. So the good thing about FOXG1 is that it's actually a tiny gene. It's so easy to deal with. It's a good thing. So this gene that I was working on is so big, and we didn't even have the tools to knock it down. So like a lot of things that are working for FOXG1 now, we couldn't even get that to work for this gene. And so what I came across is this targeted protein degradation, which came out of, which is a technology that was developed by Greg Crew's lab at Yale. I think he's at Yale. And so this was back in 2014, and I think they went on to set up a company, and now that's sort of sprung up into a whole field of lots of different companies and lots of different labs that are involved in basically modifying ways of specifically targeting within the cell. So not delivering things like gene therapy, not manipulating anything at the genetic level, but getting at things at the protein level, and doing it in a specific way. So I think a lot of what we're going to see in the next 5 years is that people are going to figure out how to target things more specifically, because we need to know which sort of, even when it comes to the cellular level, like which cells in the brain are you going to target? You can't target everything. It's not going to be beneficial for the patient to target everything. And even within the cell, which proteins are you going to, how can you sort of like control the process of doing something very specifically so that you don't mess up other things? And I think the technology is moving pretty fast in that direction.
What motivates you? What is your hope? What is your goal here? And what motivates you and gives you hope that all of our work will lead to great results for countless patients?
I mean, I think as a developmental biologist, I'm extremely excited about understanding the human brain. I think we're sort of in a new era where we can actually understand the human brain. And we have the tools to be able to study the human brain. And once we have gained more understanding of really what makes us uniquely human, we can actually use this information to cure a lot of neurodevelopmental diseases, including FOXG1 syndrome. So all of it is going to stem from understanding the dynamics of the human brain better.
Why is FOXG1 a viable gene for biotech investment? What makes FOXG1 an attractive disorder for biotechs to take notice?
Well, I think first of all, the biggest strengths of FOXG1 is actually the foundation, and you guys who have done such an amazing job at getting so organized and organizing the patient data, getting the assets. And so it's really like sort of a very rich portfolio. And the other thing is that there's a lot of effort in doing sound science that is reproducible. And so what biotechs are interested in is a problem that has an audience, which obviously you guys have because you managed to get organized, and you have so many patients collected already. And the other thing is, can it be targeted reproducibly? And I think the science that's being done within the foundation is doing a really amazing job at sort of doing rigorous work, so that at the end result will be a slam dunk for a biotech company. So once something is identified, it's not going to be very difficult to convince anyone to take this on board, I don't think.
That's helpful. Thank you for those kind words for our work. And again, we're just so happy to have you here as our Director of Science. We could do more together. And thank you. As always, we'll continue to do these updates, and we're excited to when we could start telling our community about hits.
Thank you. Thanks for having me.